Namaste to all. Welcome to the new session on drug designing and drug discovery. So in this session, we are going to see about the active site finding as well as the protein docking part. So what is meant by this? See, in any of the particular three-dimensional structure, or you can say like a motif is present or a particular active site is present, what we can do is we can create some, or we can create some molecules, chemical molecules, or we can have some protein molecules in the form of like enzyme, which will bind at this active site for knowing the particular interaction, or you can say that how the energy is there in that particular location. Everything can be taken care at this particular point here. So if you see the protein docking, so proteins, if you see in general, proteins are the functional unit of the cell. So what they do, they are the polymers of amino acids, maintain a key role in the intra and the intercellular processes. So from head to tail, from every part in our body, it is maintained by proteins only. And maintain the control functions as enzymes, because you know that for each metabolic activity or for any of the synthesis, enzyme act as a catalyst, and they are being a part of this proteins only. So if you see the characteristics elements, they are like a carbon, hydrogen, oxygen, nitrogen. So these are the characteristic elements which are present in this protein or polypeptide chain. And when we are designing or when we are framing the protein molecule or protein chain, the main important part is the CN bond, which is a peptide bond. And also along with that, we are having the CO and CH and N, like that, bonds which are being attached with other molecules here. So if you see the definition of this docking, this is actually called as to place a ligand into the binding site of a protein in the appropriate manner for the optimal interactions with the receptor protein. So as I said, we are making the ligand. Okay, ligand means a particular chemical molecule. It can be like a synthesized proteinaceous molecule, anything it can be. So to place that ligand into a binding site. Binding site means a place where it is having activity, where it has the angle, where it has the degree, everything. So this is like an active site where this ligand will come and bind, so that the optimal interactions when they takes place with this receptor protein, they will have their action now. So if you see the goal, what is the goal in this is to be able to search a database of the molecular structures and retrieve all the molecules that can interact with the query structure. So that is what is our main aim is. That is, we are having our query structure, which we have got from various prediction tools. And after getting the prediction tools from that, what the molecule has got, the 3D structure molecule, what we have got, whether they can bind or interact with this particular active site. That is what is our goal is now. So the protein-protein docking, we are going to speak about two different kinds of docking here. First type is we are going to discuss as protein-protein docking. The second type is the protein-ligand docking. So if you see the protein-protein docking, the aim is to predict the structure of the protein complexes from its partner. So what we are having is we are having two different structures, and based on the article of James Miller in 2007, they say that the monomers which are present when they join together by some interaction or by some forces, what happens is they are now called as complex. So this is how the docking study actually starts, basically to say. So the types of docking studies, as I said, there are two types of docking studies are there. The first type is called as a protein-protein docking. So protein-protein docking means what? That is, both the molecules usually considered rigid, means very strong. They, which is according to their angle, according to their torsion, everything, they are very rigid. So the 6th degree of freedom is there, and such space and the energetics of possible binding conformation. So protein ligand docking, if you see, these are actually flexible ligand and rigid receptor, basically to say rigid receptors. So what they do, they reduce the flexible ligand to rigid fragments connected by one or several hinges, or search the conformational space using the molecular dynamics. So what is molecular dynamics? Basically, it talks about various things like what is the thermodynamics aspect is there, what is the heat which is being released, or the angle which is released, or torsion which is present. So everything is being spoken about this molecular dynamics. And in this, the conformational space, what is there? That is, conformational means in which angle it has to turn so that the space will be visible. So all these things is seen mainly in the protein ligand docking.